Hi guys, and welcome to my first in my series of clinical biochemistry videos. And in this video I'll be giving you an introduction to sample preparation and some of the factors and influences that can happen to the sample quality, thus affecting any patient results. So the learning objectives for this is to describe what reasons a clinician would need to collect a sample and what types of sample they would collect, to describe what factors may contribute to sample variability, to know the difference between plasma and serum, and believe me, there is a difference, to outline some pre-analytical problems with blood samples, and then to be able to apply this knowledge to case study situations. So this is once you've gathered the basics, you are then able to use this knowledge to help in real life situations of what's going on. Okay, so this first part I'm just going to ask you guys, so you can pause this video as well. What reasons and types of samples do you think a clinician would like to get? So what reason would, say, not just necessarily a clinician, but a researcher, doctor, or anyone in general, so what reasons would they like to collect a sample? Okay, so after you've had a moment to think, so some of the reasons that they would probably like to get a sample is probably for diagnostics reasons. So quite often when you go to a hospital and you need any tests, they will need these for diagnostic reasons. They're not just doing this just because they want to have parts of you as like some sort of safekeeping or anything like that. They're doing this for your health. Okay, so these are getting your samples for diagnosis, be it blood or whatever. Okay, often they will need it for research. So this is quite often being used in cases of people suffering with cancer. So certain cancer patients would be exposed to clinical trials or certain medication, and they would like to investigate how these drugs are then aiding in their cancer treatment or how it's affecting it in general. Preventative measures, so this is sort of like screening, so checking if anything could potentially go wrong. So these preventative measures could be done mainly for like genetic sampling or stuff like that. Monitoring patient. So imagine we have a patient and we are giving them a course of medication. We're not just going to give them medication and say, right, that's it, you can go home and do whatever. No, sometimes you'll need to keep them and you'll need to be constantly monitoring to see whether the treatment is being successful or not. Because quite often, if you do have some treatment, it may not be 100% successful. So if you're given this drug and it is not successful, you could go home and die. So the clinician needs to monitor you to make sure, yes, this is working, and if it's not working, we'll try something new. And then as I get screening. Okay, so the next question is, so what types of samples do you think can be collected? I've given a few as examples, but there are actually dozens of different sorts of samples that can be collected. So just cover for like a few seconds just to think about what types of samples can be collected. Okay, so some of the types of samples that can be collected are blood. So blood is one of these obvious ones. Okay, so there are dozens of different blood tests you can do. So you're checking like the glucose levels, potassium levels, any sort of electrolyte levels, hormone levels, hematocrit, so checking all the relative blood levels or anything like that. Okay, again, plasma, so that's the liquid component of blood, and serum. So in a minute I'm going to tell you what the difference is between plasma and serum, because I gather that most people probably think that they are pretty much the same, but they are not. There is a slight difference, and I'm hoping that after I discuss this, you'll probably know the difference. Okay, so then there's urine. So obviously if you've got urinary tract infections, we need a urine sample to test the presence of proteins in there or any form of toxins in there for toxicology reports. CSF, which is cerebral spinal fluid. So this is checking to see whether all the cerebral spinal fluid in your brain is all safe. So this could be a test for meningitis, for example. Saliva, so you can literally just spit into a tube and then you can send this off. So this happens like if you're screening for blood. So for example, I did some work where I was helping people sign up to be bone marrow donators, and what we did, we got them to spit into like a little saliva tube. We send the saliva off to a lab and then they'll test it for bone marrow compatibility. Okay, then we've got sputum. So some of you might think that saliva and sputum are the same thing, but they're not. So saliva is literally what you've got in your mouth. Sputum is what you've got in your lungs. Okay, that's the fluid component in your lungs, so like your phlegm or like that. So sweat, as you know, we can test sweat for different things. So you can test for like salt concentrations, presence of bacteria, etc. Semen as well, so you can So often, like when people are donating semen, for example, for like sperm donations, they'll analyze the sperm to check the quality of the sperm, the number of sperm cells per teaspoon or per mil, or however they're measuring it. So pus as well. So if you've got some sort of infection, they'll analyze the pus. So this is often the case for things like necrotizing fasciitis. They'll get an example of this lesion, which I've just mentioned, all the tissues. They'll do microscopic tests on it to check the presence of Streptococcus pyogenes, for example, and then giving you diagnostics of necrotizing fasciitis. You can then also send in feces. So this is quite often for people who are suffering from colon issues, especially like colon cancer. For the presence of any blood in your feces can then go on to help in the diagnosis of colonic cancer. Areas of infections, as I mentioned, like if you've got pus, or even if you've got like a sort of rash or anything like that, you can swab this area of infection. Again, run it under microscopy and see what there is there. And also just general swabs. So even like when you're doing your cervical cancer screenings, so they'll take a swab of the inside of the vagina, then they'll send us off to labs for screening. Okay, so now we're going to talk about the difference between plasma and serum, okay? So, in whole blood is consisted of three things: plasma, a buffy coat, which is your white blood cells, and your blood cells, okay? So the white blood cells and the red blood cells are what I call your formed elements. Okay, so these are your cellular components. And plasma is purely the liquid component of blood. And then your usual blood is about 55% is plasma. So this is the fluid component of whole blood, and it contains anticoagulants, and it must contain, anticoagulants after centrifugation, because plasma can still contain molecules which can aid in coagulation. So these will need to be chelated and removed. So for example, use an EDTA or citrate. And serum is the undiluted extracellular portion. So again, it's similar to plasma, but this is after coagulation has occurred. So in this case, you're letting the blood clot, and then the fluid component that's released is the serum. Okay, so now we'll start talking about some of the factors affecting samples. So these are things which can either affect the quality of the sample or your ability to collect the sample in the first place. So for example, might restrict you collecting it, might restrict you in storage or whatever. Okay, so we can divide these into two broad categories. We've got endogenous and exogenous factors. So endogenous factors are those which we cannot change, okay, or are very difficult to change, okay, so we cannot manipulate these in any way. Then you've got exogenous factors. So these are things which we can change, so we have a degree of manipulation which we can do. Okay, so just have a moment to think about what sort of endogenous and exogenous factors there are which can affect your sample quality or collection. Okay, so let's just see what sort of examples you came up with. So here's a list of examples of endogenous factors. Okay, so we've got things like genetics, age, body mass, sex or gender, allergies, fitness, stress, race or ethnicity, pregnancy, stage of the menstrual cycle, or the prevalence of any other underlying conditions. So probably immediately straight away you can tell that some of these we cannot change just yet. So things like genetics. So at the moment we cannot change someone's genetic makeup. Okay, we can cause mutations, but we can't fix genetics in a way for humans, okay, because it's not ethically approvable. But there is recently technology involved in CRISPR-Cas9 technology, which can actually change the DNA to become like more healthy. So this is currently being investigated for things like cancer cells, so changing the mutation in a cancerous cell to restore it back to a healthy cell. And again, age, we can't change someone's age, okay, unless you build like some sort of magic time machine to send you back in time. Then you've got something like body mass. So body mass you can change, but it's quite difficult to change over a prolonged period, well, over a short period of time, should I say. Because this is something that requires extensive amount of time to do. So you can't all of a sudden go on a diet and boom, lose 10 pounds in one day, okay? You can't do that at all, okay? Unless you're literally putting your life on the line. It's quite dangerous to do that. There are other things such as stress. So stress is a really important factor for sample quality. Okay, so under stress, your body can release epinephrines, endorphins, and adrenaline in particular. So these can affect how your body's reacting to certain things, okay, in that fight and flight response. Even like pregnancy and stages of your menstrual cycle can affect something. So in your menstrual cycle, your hormones will be all over the place throughout the month. Okay, so certain times of the month can affect your hormone levels and how your body can respond to certain things. Okay, so now we're going to move on to exogenous factors. So what sort of exogenous factors do you think can affect the sample quality? Okay, so let's just see what sort of examples you could have come up with. So there's quite a few examples which, again, can affect your sample. So patient preparation. Okay, so how the patient is being prepared prior to investigation. So for example, whether they have to drink a certain fluid for like dye control. So like when they're doing like radiography, certain radiography tests require you to analyze the blood flow, and like barium dyes, for example, can be used as the absorbers to test the flow through blood vessels. The actual sample collection itself is an important factor. So imagine if you're collecting like blood, for example. If you don't collect enough blood, you're not going to be able to run any sufficient tests. Okay, so sample handling is another thing. So imagine if you've got good quantity of blood, you've got good quality blood. If you don't handle that in an appropriate way, that whole sample could be literally destroyed. So literally like putting it in the wrong container, for example, which I'm going to talk about in a minute with vacuitainers. So these are little tubes which blood gets put in prior to testing. So this can like affect the coagulation or how the blood behaves for certain tests. So medication is another thing. So whether the patient is on any medication during the time of analysis, so you can change the medication easily and then do further tests on that. So this can also be related to timing. So again, as I said, stage of the menstrual cycle, it's hard to control exactly where it is, but timing of certain things. So for example, collecting sample and then putting it in the fridge could have like a minute time span, for example. So you can easily control that 1 minute time period. Same thing for temperature. So you can either go from like a warm room to a cold room to like store your sample. Okay, so this is just a picture of some of the vacuitainers you can get. So these are color-coded tubes, which can either be plastic or tube, and each of them will contain a different item or additive. So one of the examples that I'm going to just talk about is this lilac one, or lavender, however you see it. So this one contains EDTA. So EDTA is an excellent chelator. Okay, so this will form complexes with ions, forming a complex, as I just said. And what this is useful, so in your blood you have free-flowing calcium ions, okay, and these are essential for coagulation or starting the coagulation process. So what EDTA does, it will bind and chelate to these calcium ions and it will prevent these from initiating coagulation. So therefore this is acting as an anticoagulant. And again, each of these tubes contain many different things and they're each used for different things. So as you can see here, this red one contains serum, for example. What's used for serum? Okay, so just some more things that can affect your blood sample. It depends on the patient posture. So for example, whether they are supine, which means laying down, sitting, or lying. Okay, so your plasma volume can decrease by 14% from supine to standing. So when you're laying down, your blood plasma will be significantly higher than when you're standing. Okay, so this can affect your electrolyte concentrations, which can either give you false negative or false positive results. So tourniquet. So tourniquet is the band that goes around the patient's arm, and what this does, it prevents blood flow down the arm, so it causes like the veins to bulge out, so it's easier to get the needle in front to get a blood sample. And this can cause like a loss of water or electrolytes to the interstitium, so again, it can give you false negative results if it's not done carefully. And as I say, quantity. So if you get the incorrect amount of a sample, it can seriously affect your results. So for example, in one of the vacuitainers, it contains citrate, which again is a chelator, so it can bind to calcium to prevent coagulation. And some of these need to be done in a 9:1 ratio of blood to citrate. But there's also another citrate tube which has to be done in a 4:1 ratio, so you need to make sure you get the exact ratio for these tests. And also the quality of the sample itself. So if you're collecting blood, you don't want it to be hemolyzed because you want to analyze the blood itself. And then also if you've got any IV fluid contamination, which can actually dilute your sample, so again, it can give you false negative results. So So just going to talk a little bit more about haemolysis. So haemolysis is the destruction of red blood cells, and this will release your red blood cell content. So that's all your hemoglobin gets released into the blood. You've got all like the electrolytes within the cell itself, such as potassium, can be released into blood. So if you've got haemolysis, levels of potassium will be higher, so you can get false positive results for potassium concentration in the blood. But again, some of the enzymes which you can find in there, or proteins, include aspartate aminotransferase, so AST, and alanine transaminase, ALT. So these are two proteins which are associated with liver function tests. Okay, so if these are present in your blood, that's an indicator of whether you've got like any form of liver disease. But it could be this could be a false negative result. If you've got hemolysis just purely because of your sample, it's going to be an indicator of liver function. So you might be trying to assess your patient for any sort of liver function, but they're not really suffering. It's just the quality of your sample itself. And again, lactate dehydrogenase could be an indication of tissue injury. You may not be suffering from any tissue injury, but because the red blood cells are hemolyzed, it's released this into your blood. Then again, as I mentioned, when red blood cells are lyzed, they can release their electrolytes, so like potassium, cholesterol, and phosphate. Okay, so just using everything we've just sort of gathered throughout these few slides, let's just apply this to a case study. Okay, so we've got a 71-year-old male with chest pain, shortness of breath, and was placed on IV infusion. The clinician recommends a blood transfusion as they have a critically low hematocrit and hemoglobin. So after this first sample, they then took another sample, repeat, but this time using a venipuncture. Okay, so if we just have a look at the first sample, so we've got a hemoglobin of 4.7, but if you look, the ideal reference range is 13.5 to 17. So that is drastically low. So you might be thinking, oh gosh, this person is suffering from anemia. Again, you can tell from the hematocrit. So that's your relative percentage of red blood cells. So usually this is about 42, 44%. Well, again, as it says here, 41 to 53 for this sake. Again, because this is so low, you might be thinking, oh god, they've had like serious blood loss, for example, so you don't know what could be going on. Sodium levels are within the normal range, but again, they're on like the high range, so you don't know whether something's going on. And again, for like chloride, for example, again, it's high, but this time it's outside of the reference range. So you might be thinking, why are the salt levels high but everything else is low? So you might be thinking it could be excessive salt, for example. But then you've got like potassium levels, which are again critically low. So you can sort of start piecing together how these and the reference range. So you can see that most of these are a lot lower than the reference range. However, if you look at sample 2, it's higher. So this is just taken just a few minutes later. But again, you can see how the samples are now a lot closer to the ideal range. So what has happened here? Just have a moment to think, what has happened here to cause this variation in results? Okay, so what has actually happened here is that during the first sample, when the man was on IV infusion, this has diluted his blood content. Okay, so everything in his blood was diluted. Everything in the sample was diluted. The reason why sodium and chloride levels are elevated is because the IV will contain a natural source of salt, hence why these appear relatively normal while everything else is low. And then after the second sample, so this has got less dilution, but you can see it is a lot closer to the normal value. So this is a good looking set of results. But just think what could have been the consequences of if the clinician did go with these results. Okay, so the clinician recommended a blood transfusion. So this patient could be receiving an inappropriate blood transfusion, which could be dangerous considering their condition, or it could be just a waste of resources, for example. So it's really important to do repeats if there are any uncertainties going on, so you can actually figure out what is definitely going wrong. Okay, so now we're going to move on to the test yourself section. So here I'm going to ask you a series of questions for you to answer. I'm not going to give you a mark scheme, and I'm not going to tell you how many marks each question is worth. Okay, I'm going to leave you to figure that out. Okay, so the first question is, what reasons would someone want to collect a sample? What samples could possibly be taken? What factors can affect the quality of a sample? And here for a case study. A female patient is wanted to test to see whether or not she is suffering from an overactive thyroid. Upon blood sample analysis, her thyroid hormone levels were all normal. So that's already strange. So what sort of sample collection errors could have occurred in order to produce a false negative result? And then upon further examination, it was shown that the patient is not suffering from hyperthyroidism. In the event of the false negative result, what could be done in order for the scientists to be certain of their results to ensure a proper diagnosis? Okay, so this has been a video on about sample collection and factors affecting sample collection and making sure that you've got a good sample. So again, guys, thank you for watching. I'm happy to be back making these videos. It's seriously helping me, and I'm hoping it's helping you guys as well. And as always, leave a like if you have benefited from this. And also, if you've got any questions, feel free to drop them in the comment section below, and I'll get to you as soon as possible. And also, if you want to, why not just add questions of your own for other people to answer? So make up your own case study, like I've done here, for example. Just try and get just people involved and just try and like aid your learning, for example. It will help. So as always, guys, thank you for watching, and I hope you guys enjoy. Peace out, guys and girls.